Good evening and warm welcome to this webinar hosted by Universal NutriScience in collaboration with International Human Embryology Research Academy. I think most of us are trying to figure out why our portfolio of gold and silver is falling. We may or may not figure out the reasons for the same, but today we have international as well as national faculty which will help us to decipher the role of tiny messengers, the role of extracellular vesicles in ART. I'm Dr. Manish. I'm the Medical and Regulatory Affairs Head at Universal NutriScience, which is collaborating with the IHERA for this webinar. And I'm Medical and Regulatory Affairs Head from UNS. UNS, as we all know, is a pioneer in nutraceuticals for last 25 years out here, helping fertility specialists manage their patients well with nutraceuticals like coenzyme Q10 and our recent launch of resveratrol. So with this, I'll request Nakul over to you for your welcome note to the faculty. Nakul is our marketing lead for our men's health portfolio. Nakul, over to you.
Thank you, sir. Thank you, Manish, sir. Good evening, everyone. Warm welcome to each one of you in our UNS IHERA webinar. It's true pleasure to see many esteemed IVF gynecologists and embryologist experts from across the world who are gathered over here on this platform. Everyone is a composer and inventor in the field of reproductive science, who are the consistently innovating, advancing the new techniques and contributing to the medical breakthroughs, and that are the transforming lives across the country. We at UNS, we take immense pride in the being one of the fastest growing nutraceutical company, which is dedicated to making the meaningful impact through science backed high quality products. And our infertility portfolio, which is CoQ 300, CoQ LC, CoQ Ford, CoQFem, and CoQMAN, which is crafted to empower individuals and support their journey towards the parenthood. So with this, I'm my honor to invite today's coordinators, and they can proceed to introduce our today's speakers and chairpersons.
Thanks, Nakul. So today we have coordinators for this webinar, Nidhi Madam. She is, and we also have Yoshita Tanwar Ma'am. She is a senior clinical embryologist. Nancy Sharma Ma'am, she's a senior consultant embryologist. And Anantha Lakshmi Ma'am, she's a consultant and senior clinical embryologist. With this, I'll request pass on the dais to Nidhi Singh Ma'am for further proceedings. Ma'am, over to you.
Thank you so much, and a wonderful and a warm evening to all of those who have joined us today. We have a very insightful scientific webinar taking place today, in which we will be entering the microscopic world. As it is said, big breakthroughs often come from tiniest messengers, and today we will explore how the extracellular vesicles are reshaping our ART. And for this, and I can have the next slide, please. I would invite our founder, Dr. Ved Prakash sir, our co-founders, Sanjay sir and Charudha Joshi sir. And I request Charudha Joshi sir to give us a welcome note.
Thank you, Nidhi. Very good evening to all. And on behalf of IHERA, I would like to invite and thanks all of you to be part of this event today, joining this fantastic event in IHERA. This all has been started along with the giving platform for the new scientific developments happening and to discuss various things. Right now, we also know that cell and cell organelle and process and mechanism, still it is not very well understood. And there are lots of research activities going on, and there is a lot of things yet to come. So here in IHERA, we started this series of webinars with the aim that providing platform for interacting and discussing the latest developments. And as Dr. Nidhi rightly said, this life starts with a very small and tiny structures, and then it ultimately converts into a full-grown life. So for this, today we have this webinar on role of vesicles in ART, and we have expert international speaker, Dr. Vago, And we have our Indian speaker, national speaker, Dr. Krishna Chaitanya. So let's see. We expect to hear what is the latest development happening in the field of this ART related with the vesicles, its role, and further for further things. Again, hand it over to Dr. Nidhi. Once again, on behalf of IHERA, I welcome you all for this webinar. Thank you.
Thank you so much, sir. Can I have the CV, please? So beginning the session, I would like to introduce our first moderator. Again, we say that ma'am very rightly said, don't need introduction to be long. Ma'am does not need a long introduction. Everybody knows Dr. Bindu Chamote ma'am here. She's a wonderful person. She's an IVF scientist and consultant clinical embryologist. Welcome, ma'am. And I would request you to please introduce her.
Thank you so much. Warm welcome to everyone. Good evening, and maybe still a good afternoon to our international speakers. Maybe mid-afternoon, 2:30, you are still out of your lunch time. So at the outset, I would really like to thank IHERA for yet again coming up with such a wonderful topic. It's a very basic topic and yet not a routine one. Something out of the box, something which we really need to understand. And communication is the key, whether at the macroscopic level or microscopic level. So communication, signaling, and regulation of that communication. And that is where extracellular vesicles come into play. So they are the ones that are regulating the whole scenario. As rightly said, they orchestrate life. And who better than two wonderful speakers here to speak about the role of extracellular vesicles. Personally, I would say that I had restricted knowledge just related to biochemistry, what are extracellular vesicles, and maybe how they help from follicular genesis to implantation with respect to oocyte and embryo. But to hear today from our eminent speaker, Dr. Vago, about how they help in the sperm motility and capacitation, that would be something. So with that, I would like to introduce, please, the CV, please. A very interesting topic that we all are going to hear from Dr. Vago. Very heartening to see that we have a very dedicated group, and Dr. Ricardo Vago is a group leader in extracellular vesicles and toxin group, Urological Research Institute at Milano, Italy. He's also an adjunct professor of applied biology and genetics. And he has more than 20 years of expertise in molecular oncology and drug delivery, expert in the development of toxic-based therapeutics and extracellular vesicle nanodevices for personalized medicines. So yes, of course, we are talking about here nanoparticles and micro level, the micron size particles here. So over to you, Dr. Vago. We would love to hear what you have to tell us about this topic. Thank you. Welcome.
Thank you very much for inviting me for the presentation. Now I'm gonna share my presentation. Okay. Thanks again for inviting me. Very happy to be here to share our data with you and to discuss about this topic. Extracellular vesicles are very strange things, but are very wonderful. As you were saying, they are a system that your organism use to communicate among cells, among tissues, all around the body, like the nervous system, like endocrine system, and many others. In this case, we are focused on the role, the impact of the extracellular vesicles from different regions of the female reproductive tract as they promote spermatozoa motility and support capacitation. It's a funny story, those of extracellular vesicles, because in principle, they were considered to be only garbage bins. So for decades, they were thought only that cells can waste everything that they do not use. They can be potentially toxic in these vesicles, and they secrete and waste away. But seminal works pioneering works in the early 90s by Jan Lotwal and especially Grazia Raposo demonstrated that, for example, B lymphocytes secrete antigen-presenting vesicles. So vesicles are cooperating in a lot of processes, physiological processes in the organism. So they started to be interesting object, and they started to increase the interest around them, and then the publication starting from 2000 and more. In 2011, the International Society for Extracellular Vesicles were founded. And years later, also national societies were founded. In 2018, the Italian Society for EVs, and then in a few years later, the Indian Extracellular Vesicle Society. So what are EVs? They are objects surrounded by a lipid membrane that can range between a few 30 nanometers, 50 nanometers until 1 micron. So it's in the nanoscale or in the microscale. They are present in all the body fluids, as we will see in a while, as well as conditioned cell culture media. And they can be divided not for the size or the shape, but from the biogenesis. They are released by secretion of multivesicular body contents, for example, and these are the smallest, and they are called exosomes. Or they can be produced by plasma membrane budding. In this case, they are called microvesicles, or they are originating from the formation of apoptotic bodies during the programmed cell death. So they collectively are called extracellular vesicles. They are observed in health and disease conditions, and really can be involved in many different processes inside the organism: coagulation, healing, proliferation, remodeling, immune regulation, as was saying before. But they are also part of the disease development and progression. For example, in case of cancer, they are known to be increase the tumor spreading, but also the angiogenesis and the degradation of the cellular matrix. So they are like, they have two different sides of the coins. One is good, the other one is bad, like the Latin god Janus. They are present in all these body fluids, starting from CSF, but also breast milk, blood, especially seminal fluid, uterine fluid, urine, and so on. So in male and female. So they can be collected in all this body fluid, found and collected. They have really a lot of potential, since they are mirroring, they are resembling the cell of origin. They can be used, as example, in liquid biopsy for diagnosis, prognosis, or monitoring the therapy for predictive purpose. Or since they are part of our body, they are biological active, and they can be also exploited for therapeutic purpose as drug delivery systems. So they have a really great potential that they can have in the future studies. But just go to the focus. The long journey of spermatozoa starts from the male reproductive tract, especially in the epididymal lumen. The epithelial cells are secreting vesicles. They were called epididymosomes. They can be captured by exosomes and can induce sperm maturation, can support sperm motility, and can also prepare spermatozoa for acrosome reaction. So this very long trip starts and goes on to the female reproductive tract, continues, where the efficiency of in vitro fertilization by improving parameters such as viability, motility, capacitation, and finally binding. You can see here how many parts of this male reproductive tract are producing vesicles. So the endometrium, for example, the follicular fluid, uterine fluid, and so on. So let's start from the beginning. We studied in the last 10 years the impact of vesicles on spermatozoa. Was known from study in animal models that proteins associated to epididymosomes, so the vesicles produced by cells in epididymis and epithelial cells of the epididymis, can be captured and can transfer protein, active protein, to spermatozoa. And this was one of an important part of the spermatozoa maturation. So we started to characterize this kind of vesicles isolated from the seminal fluid. We started by analyzing seminal fluid from normospermic men and compared with astenospermic men when spermatozoa have impaired motility and they are not so efficient in fertilizing, in reach and then fertilizing the oocyte. So we collect seminal plasma from 11 individuals, then isolate extracellular vesicles. 6 were astenospermic men, 5 normospermic men, extract protein, analyzed by mass spec, and through database research and bioinformatic analysis, we revealed more than 2,000 different proteins into these patients that can differentiate these patients. So to make short a long story, some of these, one of the highest, more present protein in the astenospermic man was glycodelin. While on the other hand, CRISP1 was in the group high represented in the normospermic seminal fluid. We validated this data into patients independently, and we saw that both glycodelin is confirmed to be upregulated in the astenospermic man, while CRISP1 was upregulated in the normospermic man. Other proteins like TSG101 and flotilin that are typical of extracellular vesicles are present in the same amount in all samples, as well as the prostate specific antigen as housekeeping control gene, control protein. Glycodelin is known to be a negative modulator of sperm function because it inhibits albumin-induced cholesterol efflux from spermatozoa, and so in this way it may prevent capacitation. So we analyzed the vesicles from 60 individuals in order to measure the amount of glycodelin, and we saw that it's significantly increased into astenospermic patient compared to normal patient. So in this case, we have highlighted the presence of a negative an inhibitor, a negative regulator of sperm function that is present in extracellular vesicles. On the other hand, CRISP1 is an ion channel overrepresented in semen vesicles from normospermic men, and it's a key regulator of calcium efflux that once inside the spermatozoa is able to trigger a series of events that leads ultimately to the activation of phosphokinase A, and then to the hyperactivation of the tail and the protein tyrosine hyperphosphorylation in order to sustain to trigger the sperm motility. We have found that this protein can be, these are the results in Western blotting, is able to be transferred through the extracellular vesicles to spermatozoa. So we have here enriched the spermatozoa after incubating them with EVs enriched in CRISP1. Another interesting thing is the percentage of uptake by spermatozoa. In this case, we tested the kinetic of capturing by spermatozoa of stained EVs after 15, 1 hour, and 4 hours. We have a different percentage that is due to the donor, 6 different donors ranking from around 5 to 15%. And another important thing is that vesicles can be higher taken up by time. 4 hours are better than 1 hour, but also are dependent on the pH. So neutral pH 7.5 is a little bit worse compared to acidic pH around 5.5. And this could be important implication, especially in the female reproductive tract, where the pH is changing depending of the tract considered. Then we investigated what kind of internalization is mediated by spermatozoa for EVs. Is an active process because it's impaired at low temperature. And then we tested different inhibitor of this process. And then we saw that the philippine, that is inhibitor of caveolin-mediated raft mediated endocytosis, can reduce in a dose-dependent manner the uptake by spermatozoa. So the conclusion on this first part just can be elucidating. This evidence our finding can show that seminal EVs can carry proteins can potentially are able to favor or like RISP1 or inhibit the reproductive process in humans. Mature sperm cells can still receive EV-derived protein cargo after ejaculation, mainly via lipid rafts mediated endocytosis. Then motility and the ability to undergo capacitation can benefit from EV transfer. And also semen quality is affected by male tract extracellular vesicles. Now move to the second part of the journey through the female reproductive tract. So this way, and this was the last paper that we published last year, we collected follicular fluid, cervicovaginal fluid collected at day 2 and 7 after the LH surge, mimicking 2 different phases, so ovulation and the window of implantation, and then the spent medium of this decidualized and not decidualized endometrial stromal cells. So all these, we isolate EVs from all these fluids and then investigate the impact of co-incubation with spermatozoa. The uptake, starting from the uptake, going to the sperm vitality, motility, and capacitation. Let's start from the characterization of extracellular vesicles from these different regions. The International Society of Extracellular Vesicles gave some guidelines in order to have a good characterization of EVs that include the determination of concentration and size. In this case, you see it's more or less the same for all our samples, between 150 nanometers and 170. Then morphology, and this is transmission electron microscopy pictures that show that vesicles are around 200 nanometers or less, and they are very similar in all our samples, so the classical cup shape morphology. And then the expression of typical proteins like ALIX, TSG101 that are typical of multivesicular bodies that are where the exosomes are originating, and CD63 and CD9 are tetraspanions characteristic of extracellular vesicles plasma membrane. So they are bona fide EVs from in our hands. Then we measured the capture by sperm cells of all our extracellular vesicles, and in this case we have different amount that are not really exciting, going from maybe 1-2% until 5%. but we have to consider that spermatozoa are billions. So also only 1 or 2% means millions of spermatozoa that are positive in vivo. There is no great difference among the uptake by cells from our EVs. Then another important thing to determine is if EVs can impair sperm viability. In this case, we incubate spermatozoa with all our EVs, and then we couldn't see any decrease in sperm viability upon 1 or 4 hours, meaning then they can preserve the viability over time. Then the most important thing is that they have an active role in the sperm motility, since the incubation for 1 or 4 hours, it's able to increase the progressive sperm motility in vitro. And this is comparable by the classical treatment that is mediated by human seroalbumin that is used in the in vitro fertilization techniques. So all of them are increasing at different extent the progressive sperm. And finally, all our EVs are also able to enhance, to facilitate the completion of induced acrosome reaction. In this case, we have 1 and 4 hours, upon 1 and 4 hours, our incubation, we can have the effect of HSA that is positive in stimulate acrosome reaction, but also vesicles are comparable in doing this, in getting the same results. This is in case of endometrial cells, we can also play them and incubate with spermatozoa in order to see if also during co-incubation with these 2 different kind of cells, we can have the transfer of vesicles in order to better mimic what can happen in vivo. So we stained endometrial cells, and then we co-incubate with the spermatozoa for a few hours, and then collect spermatozoa and analyze the transfer of vesicles, stained vesicles originating from stained cells. And we have around 7% of spermatozoa that in this time frame were captured vesicles produced by plated endometrial cells. Okay, so just this my conclusion. Our results strongly support the evidence that EVs released in the female reproductive tract can contribute to spermatozoa maturation during their transit, preserving sperm viability or promoting both motility and capacitation. So overall our data, our evidence highlight that multifaceted role of EVs in orchestrating various aspects of reproductive physiology, primary spermatozoa maturation. So now the relevance of this discoveries, this data, these results in terms of fertilization efficacy and reproductive outcomes remain to be established. This is really a preliminary data that could be, I hope, translated into clinical practice in the future. So I would like to thank all the people that contributed to this work from the Urological Research Institute and also from the Reproductive Sciences Lab. It's a lot of people that worked in these 10 years to get all this data. I would like to mention today Camilla and Valentina from one side and Elisa and Luca from the other side. This was really a very fruitful collaboration from basic and clinical part of the science. And thank you all for the attention.
Well, thank you so much for a wonderful presentation, very clear and very seminal work indeed, I must say. And I mean, how the extracellular vesicles are localized throughout and how the sperm can uptake those vesicles. But my question was actually what you mentioned, the last thing that you mentioned in your slide, the relevance in ART. How are we going to extrapolate these findings to our IVF setup, where we are facing a lot of problems with male infertility, and this could be one of the good causes and remedies. But then how do we extrapolate this finding to our laboratory? So that was the question that I really wanted to ask. So if you have any, you have already mentioned it remains to be established, but then are there any plans? How do you visualize the effect in laboratory practice?
Okay. So it's not really to foresee in the future, but I can try. First of all, I think that an analysis, a deep analysis of EVs can highlight which kind of molecules can benefit to increase the spermatozoa maturation and capacitation. This is the first thing. We analyzed vesicles from different sources, from seminal fluid, but also from female reproductive tracts. But if we can go outside these systems, we can find also many other sources of vesicles. Vesicles can be found in all body fluids, for example, but also outside in plants, for example, or you can imagine really a lot of different sources. And they can have different property. For example, anti-inflammatory, antioxidant, pro-capacitating. They can transfer molecules into spermatozoa in order to induce, for example, calcium influx, and then to trigger motility, for example, and induce the increase of motility and then capacitation. So the first step is to increase our knowledge about the possible sources of EVs that can really be positive in improve spermatozoa features. The second one is to find clinical settings in order to apply these vesicles. In vitro fertilization could be one of the fields that we can use. We really don't know how many molecules can be transferred and which are which are really essential for fertilization. Some are important for the binding from spermatozoa to the oocyte, some other to the acrosome reaction that is really necessary just to fuse the spermatozoa membrane and degrade that of oocytes. So once we have a good knowledge of these properties, this transfer, so we can set up really a clinical settings in order to use EVs into the in vitro fertilization. A last comment about this, if I may, is some ethical issue, because usually male couples that that ask for in vitro fertilization have some problem in fertilization, and then it's likely, as we have demonstrated for astenosospermic men, that vesicles cannot be useful in improve the spermatozoa capacitation and features. So this could be a problem if we need some vesicles outside the couple. So where we can find, and how ethical is to find from other person or other sources. So I think this could be the picture for the future. So a better knowledge of vesicles, clinical settings, and the ethical issue.
Thank you so much. So the thing is that we need to identify the sources of extracellular vesicles. We need to identify the functions, and then probably something like supplementing the media with that. I mean, that's still a far-fetched dream, but then something like supplementation of sperm preparation media or the culture media, so that they may facilitate in capacitation or binding or improved motility, whatever, I mean, depending on what function we are looking at. So yes, it is maybe far-fetched at the moment, but not impossible if you really try to move in that direction. And yes, ethical issues would be there, but then again, the benefits, we have to look at what benefits they can cause to the patients. So something can come out of this, and really very impressive finding, and very interesting study. Thank you so much for giving us insights into this, and we look to the future of extracellular vesicles. Thank you.
Okay,
Am
thank
am
you.
I
Thank you very
allowed
much.
to ask a few questions?
Definitely, sir. Definitely.
Thank you. So wonderful, Dr. Ricardo, as Dr. Bindu mentioned, it was literally a seminal presentation. I'm wondering if you could trace back the association of extracellular vesicles present in the epididymis and to the etiology of the asthenozoospermic males, why this was there. And similarly, if there was a difference in the concentration of extracellular vesicles in different women groups, because you did a lot of work on the different sources from the female reproductive tracts. So there was a difference. I mean, if there was any difference, say, for example, women from a particular age group or women from particular, I guess you have done your work on seemingly normal females. But what if they were from PCOS or endometriotic, endometriosis patient or endometritis patients or so on? So this would be a treasure of knowledge if we know those things, and probably then we can extrapolate those piece of information to therapeutic ones.
Yeah, this is a very good question, I have to say. The answer it's enough complex, but I can try to simplify as much as possible. You are totally right. The asthenozoospermic patients produce vesicles that are enriched in inhibitory factor, glycodelin, for example. So they are not useful to sustain maturation of the spermatozoa in the male reproductive tract. And once isolated, are not useful again after ejaculation to improve spermatozoa features. So this is probably constitutive. I mean, the cause of asthenozoospermia can be really a lot of different. We analyzed a reduced group of patients that are, again, producing spermatozoa. They are not classically oligozoospermic, so they have a correct number in following the OMS guidelines of spermatozoa, but they are not statistically lower amount, but they have less spermatozoa. So is something that is related to all the environment, probably where the spermatozoa are matured. Since vesicles, epididymosomes, are from epithelial cells, so I think there is the problem. I mean, an increase of inhibitory factors are loaded on vesicles, and this can impair really the maturation. So this could be really studied further and deeper because it's not easy in men, of course. Most of studies were performed in animals, bulls, for example, boars, and cats and other animals. Of course, it's not really exactly the same like humans, but there is a difficulty to do in humans. So we can infer some of this of our results, but I think the study of the environment, vesicles are mimicking the cell of origin, the producing cells. So probably the problem is really in the environment that is feeding the spermatozoa. About the women, our cohort was not so big. Was enough big to demonstrate that this kind of event was really true and was reproducible. But was not so big in order to make some subgroup of women. All our women are coming from the center of fertility for the in vitro fertilization. So there are cycle of arts, for example, in order, it's not really clear which of the two men or woman have problem during the fertilization, but we get samples, for example, from all these cycles during the art, or the endometrial cells from endometriosis, but we put in culture cells that are from healthy tissue that are again explanted because of endometriosis, but they are still, that kind of cells are selected and they are healthy. So it's really a huge, only few spot of the pictures. So we need, of course, to increase to better see all the pictures. We need to increase the number of patients, and then, as you were suggesting, just to better divide in subgroups and focus on the single variable. Otherwise, we can consider a lot of confounding factors that can affect our result.
Yes, indeed, there are bound to be a lot of confounding factors because we are talking of something that are released by the cells in the extracellular spaces. So to find the exact the localization, and then they have their effects autocrine as well as paracrine. So which cells are producing and on what are they affecting? So first, to find that itself is a challenge, and then to find out some threshold values and then move ahead in complicated pathological cases. So in normal subjects, to find that out would itself be a challenge in the first place, probably.
Yes,
if I'm getting it all right, I mean, it's really new for us also to understand.
Yeah, we are looking at the image in a mirror. So we are not directly seeing cells, but we are seeing what they are producing. So just like the image that is in the mirror.
Very true, very true. So thank you very much once again. It was a wonderful lecture.
Dr.
Okay,
Ricardo,
thank
sir,
you.
you have opened a Pandora's box for us, I would say. So, because there is a very categorical statement when we give, there is unexplained infertility to many of the female and the male partners, where we are not able to express where the semen factor is going and where the oocytes are going wrong, why the embryo is not made. And to this microscopic level today, sir, you have stated the study was so brilliant. And you can really say that we are all the thoughts are running in our mind today. And with this thing, thank you so much, sir, for the wonderful lecture and the presentation. And I invite Dr. Sanjay Shukla, sir, our co-founder, to
I'm already here.
introduce our next speaker, sir. Welcome, sir. And let's continue this beautiful journey. And thank you so much, Dr. Ricardo, for this wonderful presentation again.
Thank you.
Can I have the CV, please?
Thank you, Nidhi. And I think by the time the CV appears, let me introduce, just skip this one, Dr. Krishna Chaitanya Pavani. Let me be a little, informal. People give up doing just one PhD, and Dr. Krishna is a double PhD. So you can imagine the enthusiasm and zeal that he has in this arena of academics. So after finishing BTech from India, he went to University of Skövde, Sweden, and then he got his PhD from Portugal. Then, to satisfy his unsatiated thirst, he again went to University of Ghent to get another degree in reproduction and veterinary sciences. And then he's continuing his research journey at the Ghent University. He has also authored several papers and a member of several societies involving in the research on extracellular vesicles, also other reproductive medicine topics. He is also a wonderful author and co-supervisor and supervisor of other PhD students. I am sure they are fortunate enough to get their degrees under Krishna's guidance. So welcome, Krishna. We are eager to listen to you.
First of all, thank you, Dr. Suklaji, and also Dr. Ved Prakashi for always inviting me for this iHERA's presentation sessions. And yeah, I would like to correct a few things of the resume. I think I sent an updated one, indeed, more or less the same, but I'm currently working as a senior researcher at Kent University Hospital. I'm still continuing the research journey and the EV part and human reproduction, focusing more on human work and translation of bovine to human world. With all, I think I would like to thank also and also congratulate the previous speaker, Dr. Ricardo. He made a wonderful presentation. I think I cannot beat his presentation indeed, but he made it an excellent way. It also changed my perspective of some of my thinking towards sperm, because so far my research focus were more on the female part and the embryo maternal communication part. I think there is a lot scope on the sperm part too, which I never focused. I think it's wonderful and it was very much detailed. And I'll quickly share my presentation. Can you see my slides? Okay, I'll start. So to continue on the female part, more on the maternal and pre-implantation embryo part, how the EVs are communicating. So let's go, let's take detail. I think Dr. Ricardo covered what are extracellular vesicles and probably how to isolate them. I'll quickly go through them, but very short and brief. Probably what are extracellular vesicles? Maybe if you go back, try and travel before 1983, they are coined as garbage material, as Ricardo in his presentation he mentioned. So, but until they're coined as apoptotic bodies, until these two papers came up in 1983, and which was simultaneously published in two different journals, one in Cell and one in Journal of Cell Biology, which was apparently showing the similar things. What they found was multivesicular endosomal complexes where these exosomes are released. What you are seeing currently is like the complex of multivesicular bodies where the exosomes are released. I think these are the first authors to coin the term even exosomes. Before they are called as extracellular. After that, they're called as extracellular vesicles. And they found these exosomes are coming out of these multivesicular bodies in a cell. These cells are from reticulocytes, which are from blood plasma cells that they derived. These are the first findings that they showed that the cells that are released previously considered as apoptotic bodies, but rather they're not just apoptotic bodies, there are also other small cargo material. So ever since they published, there are a lot of research articles came up with publication, and there is also a journal called Journal of Extracellular Vesicles, where they show a graph how the publications rise up. This was the publication record of the with the terminology of exosomes, macrovesicles, and extracellular vesicles. There have been a significant improvement in terms of publications using EVs as a research line. There are a lot of articles and a lot of research still going on, and it was still high on the research. I think Dr. Ricardo covered this pretty much of the classification of extracellular vesicles are broadly classified in three types: exosomes, macrovesicles, and based on the cellular size component, they vary. The larger, medium, and extra large. So to the biogenesis of extracellular vesicles, again, coming to the biogenesis, it's not, it's still, I would say, like still ongoing research, because the exact part of the EVs, how they are released into the extracellular matrix outside the cell, how they are released is still not yet proven yet. Like there are some mechanisms they show. For example, the cargo content, that is like the nucleus release some microRNA or smaller nucleic acids that are released from the nucleus are ending up in the late endoplasmic reticulum, and then they are forming in the multivascular bodies. That's what we found in the first articles of 1983, and then they're released through the plasma membrane in form of exosomes. On the other hand, also the microvesicles, they're budding through the plasma membrane and forming a bigger vesicles. And this is one of the like communication pathway that are currently identified. Of course, there are many others to be explored, and how they are uptaken, how they are released, it's still need to be explored, because biological function and biogenesis of the EVs is varied from cell to cell. So biological function of the EVs are, as I mentioned, they are released from all kind of biological fluids, from breast milk to urine samples. You find them everywhere because they are released by all type of cells, and they release, they are as an endocrine signaling pathways. They are released into the bloodstream and they're transported to one cell to other cell. They're constant in the communication mechanism. So we know how extracellular vesicles are there and how they exist and how to isolate them and how to characterize them. It's also an important part that we need to know. So the EVs can be isolated in multiple ways. These are the briefing that I can give. There are different methods to isolate the EVs best, and there is no golden standard method to determine, okay, this is the perfect way to isolate EVs. No, you can use any kind of these methods like differential electrocentrifugation, optiprep density gradient, and several commercial protocols that are currently available, or ExoQuake and QEVIs on columns and purification kits, and a lot of other techniques are used. But it depends on what your biological function that you are aiming for, how you want to isolate them. That is important. And then, of course, there are also pretty much of characterization, how it is involved. So EV characterization is broadly done by electron microscopy, nanoparticle tracking, Western blotting, and flow cytometry. Of course, there are also alternatives for that. So for electron microscopy, you can use atomic microscopy, atomic force microscopy. For nanoparticle tracking analysis is initially designed for protein analysis, but it can be also determined for EVs because EVs are in the size of proteins. They also can be used with the determination of particle concentration. Instead of nanoparticle tracking analysis, you have also dynamic light scattering method. And instead of Western blotting and flow cytometry, you can use also ELISA. And also a new microfluidic systems can also be used to determine the EV presence. These are the standard techniques that are more widely used and to characterize the EVs. So I would like to give a little bit of briefing of what Ricardo mentioned in also in his presentation, in the later parts of his presentation, talking about minimum requirement for extracellular vesicle characterization. It's important to know of the EV characterization, because as you can see, EVs have a surface when they're released from the cell. So EVs, corona itself also have some nanoparticles, like nucleic acids can be attached, like microRNA and others, non-coding RNA can be attached to the surface of the EVs. And also the cargo content inside the EVs can be also present. So to prove the presence of the EVs, we have to determine the plasma membrane proteins like CD9, CD63, and CD81, and cytosolic proteins like TSU1, CU1, or LX. Or these are the proteins to be determined to show, and these are the minimum requirement to characterize the EVs. And also for betterment, there is called EV Track, where you can search. It's a website platform developed from our collaborator, who is Anne Hendricks group. They developed this EV Track. If you if anyone wants to start working on the EVs, I think this is the best platform to check on to see whether your research line is there or not. You can just log in and see, for example, I want to find some follicular fluid EVs. So you can type it there, and then you can find information to relevant articles that are published there, and you can look for informations there. And you can also contact the reproductive task force of ISEV, which I'm one of the member of this. So you can always email if you want to start working on the EVs. We can be helpful in providing more information on how to isolate them, how to characterize them. So you know how the EVs can be isolated, how to characterize them. Now we will know how the EVs are in the reproduction, which is partially covered by Dr. Ricardo in presentation already, but I give some statistics. So these are the things that in the ISEV task force of reproductive task force, we found a lot of research started happening in the EVs in reproduction. If you see, the peak is only starting at early 2000s and early 2000, like 2012 or 2011, they started, everyone started working on EVs related to reproduction. Not so much, but now there is a lot of research happening. You can see the second peak line going up. And these recent years, there is a lot of research happening on the EVs in reproduction. Of course, EVs in reproduction this is a review paper that in which they coin different terms. For example, like in sperm, they also call it as epididymosomes, prostosomes. Indeed, the ISEV, the International Society of Extracellular Vesicles, they're not in favor of using this kind of terminology, but rather to keep the terminology as EVs or small EVs or large EVs, not to be specified towards the tissue that you are isolating. It's rather to be simple also for the terminology in journal. It would be easy. But indeed, there is quite a lot of terminology being used in reproduction, like epididymosomes and prostosomes are existing before even EVs came into the picture. So to go on detail of the research line that I have developed, and also other people that are been focusing on the embryo-maternal communication through EVs. So there is a lot of research that have been happening. So the oviduct and uterus carries the maternal information, and oviductal uterine fluids carries EVs, and the embryo itself also carries information. There is a cross connection that the oviductal fluid, for example, uterine fluids that are extracted, we can isolate EVs, which there is an exchange of the small RNA content and lipids, and there is a constant communication mechanism between them. So in terms of uterine fluids, the first article came up in 2012, focusing more on the human part, where they extracted uterine fluids. So they have these patients samples of where they isolated where they get the uterine fluids. Then they used standard ultracentrifugation technique, and they also got from the same patient endometrial biopsies, and they cultured endometrial epithelial cell culture and tried to isolate EVs from them. In this, they did 3 biological replicates of isolating the EVs, and then they centrifuse it, and then they isolated EVs through the ultracentrifugation, and overall they extracted using RNA extraction. And they performed a comparison analysis between uterine fluids that they extracted and also the endometrial epithelial cells EVs, how the microRNA cargo is varied or is it common. You can see indeed there is a lot of microRNA that is derived from the EVs or common between these 2 samples, which are from uterine fluids and also the endometrial epithelial cell line culture. So showing that there is a communication between uterine environment and endometrial epithelial cells, which plays a crucial role for embryo implantation itself. So they found this potential small RNA that is present in that. There is also another interesting study where this is from Alireza Fasili's group, where they have they cultured the trophectoderm biopsies of the embryo, and they extracted EVs from that, and they cross-cultured that with the endometrial cell line. And they also showed a communication mechanism between these endometrial cell line and the EVs derived from the trophoblast cell lines. So, which showing again a cross communication of the EVs and how they are influencing each other. And the recent article that came up from Ford's group, where it's also in the it's more in a bovine study, where they try to culture the endometrium in parts, which can be used as a biosensor, where they also showed how the microRNA cargo is influencing and showing a cross reactivity between embryo and the endometrium that they culture. They potentially found these microRNAs like 128 and microRNA 129A has an influence in showing implantation in high quality embryos compared with the low quality embryos. Coming to that's what more focused on the uterus and endometrium part. Coming to the EVs derived from the embryo culture media. The first article came up was in porcine model in 2014, published by Saladin. My work was published in early in 2019. It was my first article on the EVs, I would say, where I try to isolate EVs. This is again bovine systems that we culture. In bovine embryo culture media, we culture them in the group culture of 25 embryos in group in 50 microliter droplets, and I try to isolate the EVs from that, and then supplement these EVs to the individual cultures. So to mimic the human embryo culture systems, I cultured also the bovine embryos in the single individual droplets of 25 microliters, one embryo there, and transferred these EVs. I isolated EVs from the group culture and supplemented them for the individual culture. And we saw an improvement in terms of supplementation group, particularly there was an improvement in EV supplemented group has more blastocyst rate compared to the individually cultured. So when we culture the embryos individually, the EV supplementation group has increased blastocyst rate equal to the group culture, showing that of course, the group culture of the embryos, of course, there is a debate between the group culture and individual culture. So the group culture has more EV communication when they're cultured in the group. So there is a cross communication between the embryo, showing an influence of the embryo development increase rate. When they're in the individual group, they're rather less. They don't have any communication because they're just individually cultured. So that in terms of development rate is quite low, as you can see from the individual culture. Relatively the blastocyst rate is low. But supplementing the EVs from the group culture, that means the EVs has an influence on the improvement of embryo development. We also observed that apoptotic ratio of this of the EV supplemented group was less. Another important finding that we saw was to see that we labeled the EVs and we supplemented these EVs to the blastocysts, and you can see that EVs penetration towards the embryo, and this is the stack imaging where the blue is host staining, and then the green fluorescence are the EVs that are labeled with PK67 labeling, where you can see that EVs are penetrating into the blastocysts, and they are majority available in the inner cell mass, which also shows that the EVs has a crucial role in the communication.
Sorry to interrupt, Krishna.
Yes.
The video that you showed, there was a concentration of EVs in particular area. It was ICM.
Yes, that's what I'm telling. Inner cell mass.
Yeah.
It was
Right.
more in the inner cell mass.
Yeah. Thank you.
That shows that that EVs have but this we did not further on continue with this research. How what are the possible microRNA that are present? We did not do it, but in the next article we did showed in this article where I basically focused on again the bovine model, but this time we focused on individual culture systems and try to isolate from based on in bovine, you culture the blastocyst from day 1 to day 8. Day 8 is a blastocyst stage that they reach to the blastocyst stage, where we separated the groups, the embryos that are reached to the blastocyst, the embryos that does not reach to the blastocyst, considered as non-blastocyst. We try to pool the media from this and then try to isolate EVs from it, and then isolate microRNA, differentiating blastocyst and non-blastocyst group. From this finding, I translated this research to the human system, because in humans we also since we cultured, most of the labs culture them in individual culture systems, so we want to mimic the similar setup that we set up in the bovine system to look whether we can find some potential microRNAs that are extracted from the EVs can show a pregnancy marker of a potential implantation. So for example, like you take the day 5, the embryos that are transferred day 5, we follow up on the patients which lead to the pregnancy, and we follow up on the patients which does not lead to the pregnancy. And we already collect the droplets of each embryo, and we try to isolate the EVs and try to find the microRNA from it. But the main issue that we have been facing was trying to isolate EVs from the single droplet is quite challenging, because the minimum requirement for EV characterization will be very limited when you try to isolate from the single droplet, because you are getting very few EVs that are released by the embryo, and also it's difficult to determine its concentration and its potential cargo. But we made it possible. I could not reveal much of this information right now, because this data being developed was under the patent, which was pending patent. So we made it possible isolating from the single droplet method, and we tested 106 single droplet patient samples from pregnant and non-pregnancy group, and we isolated the microRNA from these EVs from the single droplet, and then performed RT-qPCR and microRNA qPCR in it, to showing the efficiency of the pregnancy with the microRNA markers that we have identified in our previous findings, which have a translational impact. But I will go through the little bit of information here. So we have single droplet spunt media samples from 106 patients of day 5 and day 6. And these are the pregnancy outcome parameters. Initially, I was double blind, because when I started collecting, and we didn't have the information of that. And then after determining the data of the clinical pregnancies, and also the information that we generated from the qPCR information, we generated 2 data sets to see whether they match with the clinical information that we get based on the clinical pregnancy. So the prediction accuracy outcomes, I have to mark the microRNAs because they're on the patent filing. So we found 3 potential microRNAs which have different prediction pregnancy accuracies. So showing pregnancy and non-pregnancy group, the current rate of prediction pregnancy accuracies for this specific microRNA is 75%, and then the area under curve is 0.71. That is like very fair to good quality to determine a pregnancy marker in terms of comparing pregnancy and non-pregnancy group. Similarly, for other microRNA was 70%, and the area under curve was 0.69. And then for the non-pregnancy is 64.3% of this microRNA, where you can see the differentiation of false positive, true positives, and true positives and false positives in the scatter plot. And other microRNA, we have only 64%, segregating pregnancy and non-pregnancy group. Area of undercurve is only 0.5%. In terms of, we integrated the data of combining this microRNA information with the embryo quality. That means, like, usually the embryo quality is a golden standard method. The morphology is determined. So we we integrated this information with our microRNA data, and then the prediction accuracy improved at a higher level of 83%, and area under curve was 0.76 with this specific microRNA. And also for the other microRNAs was 81%, and the area of under curve was 0.72, which was phenomenally improved when we integrated the morphology data according to along with the microRNA data. So for also the for the other microRNA was 75%, under area under curve was 0.65. There is also additional information of this. We also segregated data of live births with miscarriages and live births with non-pregnancy group. And each microRNA has different prediction accuracies, and some of them have showed very good accuracy in predictions comparing live births and live births versus non-pregnancy group. I did not show this information here, but I want to limit because of the time limitations. So based on the embryo morphology, with our lab, we have 60% accuracy of pregnancy prediction. With the EV microRNA that we have identified with the microRNAs that were prediction markers was 75% of prediction accuracy, just the EV microRNA it's alone. In combining the morphology and the microRNA, we could get an accuracy of 83.3% of prediction. This was on the single droplet level where we identified it. So as in the previous presentation of Ricardo mentioned that it's translational, like when you want to take it to the laboratory level, yes, you can take to the laboratory level. There is a chance of using EVs as a biomarkers, and EVs can use EV microRNA content can be used as a biomarkers. Of this study is a best example of showing the as an implementation to the clinical research. And on other part, we also further on to studied the implantation window of endometrial stromal cells and dysregulation to see how this EV microRNA that we have identified as a potential marker of the one which showing 75% accuracy and 83% accuracy, how it is influencing. So we took this microRNA and then, you know, the implantation window of the endometrial stromal cells, we took the endometrial biopsies, and then we cultured them in the stromal cells, and then we put them for dysregulation for 24 hours. And then firstly, we want to see the microRNAs, which are commercially available, supplementing these microRNA to the endometrial cell lines could uptake them or not. Firstly, we determined that by supplementing them, the synthetic mimics of microRNAs, and to see with the imaging. So we observed the mimics that are supplemented the endometrial cell line can uptake them, and they clearly are uptaken by the endometrial cells. You could see that they are inside here. But the labeled microRNA mimics are green fluorescence, and then Hertz staining shows the nucleus of the endometrial stromal cells. And we also did a supplementation study for endometrial cell culture with 4 subject patients of biopsies. And by supplementing the commercially available microRNA mimics and inhibitors, so the mimics are synthesized they are available commercially from the companies of Kagen and Sangan Bio. You can get the mimics are synthetically designed. These are short nucleotides of 22 nucleotide sequences. The ones that we potentially identified as pregnancy markers in the EV-derived microRNA, we use the same microRNA to supplement to the endometrial stromal cells. And we used a scratch technique to see, because the endometrium, as you know, for the implantation, the endometrium is scratched and then the embryo can be entered. So to physically, to show the in vivo mechanism to the in vitro level, we used, we cultured the cells and then we desexualized them and we scratched them with pipette tip, and we took the pictures of it, and we took a picture of that after 18 hours to see the migration activity among different patient samples of these are all the fertile patient samples. These are the patients which not having any PCOS or endometritis. And we observed the migration of this endometrial cell line of this specific microRNA supplementation group. You can see that significantly we observed for all the 4 different patients. We observed the microRNA that is showing higher pregnancy, supplementing to the endometrial cell line, stromal cell line, showed highest significance in terms of migration effect you compared to the control groups. On the inhibitor level, they're intact and they don't see any of migration. But in terms of mimic, when you supplement this specific microRNA, showing a functional impact on in terms of cellular migration. And then we also did scratch analysis on the image analysis, and we observed a significant improvement in terms of this specific microRNA, showing a significant improvement in each subject in terms of area, like scratch area, and also with closure. You could see there was an improvement in terms of the control groups and also the compared to the inhibitors. And we later on performed transcriptomic analysis on these subjects, and we identified these specific genes like SPC1, NFKBA1, one of which are also involved much of the endometrial regulations, and also these genes are more in these functions, mostly connecting to the migration, cell proliferation, and cell differentiation, which again connects this microRNA has a potential influence on the endometrium itself, showing that this microRNA that is identified a potential prediction marker can show a eventual influence on the endometrial cell line, showing that this can be used as further implementation, but it also requires an ethical approval to test this microRNA for infertility patients. It can be used as a treatment in future, but of course it requires a lot of ethicals to approve. So the future perspectives, I would say, like biomarker potential can be determined. We only focused on this biomarker only on the pregnancy prediction, but we can also use it for PGT. That's our next focus that we want to do. And also we want to do more mechanistic validation on implantation and the embryo of this research. Further on, and stem cell derived EVs can be used also as therapeutics for aging oocytes, or also for implantation failures, or for embryo development RS patients. So there is a lot of potential in terms of using EVs. Further on, I would like to thank my group and also the collaborators, and especially Dr. Kelly Tillman. She was our IVF lab director, which she helped us and guided us with all the sample collections of the sponge media droplets, and also our collaborator Anne Hendricks. She helped us a lot in EV research, as she was one of the pioneer of the EV research too. So I would like to conclude my presentation. If you have any comments and questions, please approach me. Thank you.
Fascinating. Equally fascinating as the previous speaker's presentation. And now we can imagine how fast the field of reproduction is evolving, and the avenues that are coming from the basic science, research science, is opening the newer dimensions. And probably I mean, I'm already imagining supplementing the recombinant microRNAs in the transfer media and even culture media maybe, and
Yes.
infusions of cytokines and microRNAs and EVs in endometrial cells. So a whole lot of new avenues. Dr. Bindu, do you have anything to say? I'm mesmerized.
Yes, yes, mesmerizing indeed. And I was asking the previous speaker, but I think in a way you have already taken this to the laboratory. And I have to ask you, there was this slide about characterization, and you mentioned
Yes.
even with ELISA. So how can ELISA in with micro droplets or biological fluids as well? Can they help us identify the EVs?
Yeah, I think I would rather say that ELISA is, of course, it's one of the alternative technique, I would say,
Yes.
but I would say it will be very expensive to do ELISA because the antibodies will be costing you a lot.
Yes. So
You
are
can
there kits
directly,
available? Are there
yeah,
some really specific kits available to identify?
yeah. There is one specific company. I think it's a Japanese company that currently supplying this ELISA kit specific to the EV characterization markers like CD9, CD63, and can be supplemented with the ELISA kit. So I contacted this company in the past, but they're quite expensive. But I think it's a, yeah, I would say you can use ELISA as one of the technique, of course.
I mean, anyone who can afford can try to do.
Yes, they can use it. I can give
The next question
information.
is, sorry, yeah.
Yeah,
And the
please.
next one is about the use of micro droplets culture. So we usually, for micro droplet culture, we make drops of the size of 30-35 microliters, maximum maybe 50 microliters. So what is the volume that you require to be able to identify and isolate these? I mean, the volume of micro droplet, because we cannot possibly have more than 50 microliters for
Yeah.
the culture of our embryos.
Yeah, definitely. That was the challenging part that I mentioned. So we patented this isolation method because we tried different combination. The lowest that we isolated was from the ones that we also culture in our lab is 25 microliter droplets. So if it's possible with 25 microliter droplets, definitely it's possible for 50 microliter droplets.
No, when we say we need the volume of 25, that means the droplet size has to be more. And the practical,
Yes.
I'm asking this because we have done GCSF in micro droplets, and I know how difficult it is to collect, and especially with the layering of oil. So you have to be particular not to contaminate your volume with that oil. So we had to be very specific in how we pick the volume from our micro droplet. And
got it.
Yeah,
yes,
so,
I understand your question. So the tip that I can provide you is like I always practice that also for the clinicians here. For the lab, we also suggested the same. The first thing is like the filter tips we use, and also firstly remove the oil if it's possible. Remove the oil, and then you, of course, the embryo is already transferred. After the embryo transfer, just remove the oil, and then you can take the droplet out. Try to use also for the collection, try to use low binding tubes like protein or DNA low binding tubes, and try to use the filter tips. So it will help.
Okay. And one more question. So what we can also visualize is that the blastocyst itself, we can, I mean, it could be an immunological role as well here that we are talking about, because it could be modulating or regulating the immunological, I mean, balancing the immunological factors at the feto-maternal interface as well. I hope I have been able to put it right. What I mean to say is, are EVs responsible for maintaining the immunological balance? Because when we talk of implantation, we are talking about the immunological tolerance.
Yes,
right?
I
So
understand. Yeah, it's an excellent question, because the immunological, because for embryo implantation, you need to see different factors. I think immunological perspective is like to looking at the macrophages and some of these defender mechanisms of immunology, like immune system, are important. I think they also release EVs. So there is a cross connection, but frankly, I never explored in that part. I think it'd be interesting to look at that perspective too. We only focused on how the endometrial stromal cells and epithelial cells react. Maybe also the immunological cell line cultures co-cultured along with the endometrial cells, or like supplementing them. we could try that also,
The
because
only
that's also an
challenge there
important
is
part.
we cannot do it online, in the sense that in the same cycle. I mean, you can still measure the EV in my spent culture medium, but then how are you going to see the uterine or endometrial tissue? You cannot measure. So some what else can be done for that? We cannot measure in the endometrial tissue, right?
You mean the EVs from the endometrial
Yeah.
tissues? Yeah, we can do it actually. So
No,
I
for
think
a patient where we are going to transfer.
yeah, we're currently doing
Right.
that in a mock cycle. You can, like, if the patient is coming, and we can also in parallel take the endometrial biopsies of the patient which is going for IVF treatment. So if we can parallelly culture the endometrial biopsy of this patient and then isolate the EVs from the spunt media of the same patient, we can transfer that and see how the cross reactivity of the EVs of the embryo that is going to be transferred to the patient on the also and parallel the endometrium of the same patient.
Okay,
I
yeah,
think
but
I
then
think
that always is a challenge, because even during mock,
Yeah,
we do that. It is not necessarily the same case when we actually transfer,
yeah,
when we
of course.
actually do embryo transfer.
I'm only thinking of
Yes,
like research perspective. Indeed,
yes,
for the
yes.
application, it's like really challenging indeed.
I think there could be a controlled double-armed studies
Yeah,
where
double-arm studies.
you can take up the EVs and other immunological markers. We already know there are several markers, especially in cases of suspected immunological factors, the repeated implantation failure patients. And
Yes.
then you can have the pregnant patients or the previous, known fertile patients. And then we can compare, and the presence of the specific EVs or microRNAs can be categorized, and then those things can be extrapolated.
Yeah.
Yes. Very interesting indeed.
Endless.
Yes.
And Dr. Bindu, what you asked, because I worked a lot on ELISAs during my PhD. So
My entire PhD is on ELISA.
yeah, so I believe that the sensitivity would be less than the amplification, after amplification. So maybe flow cytometer and PCRs can be better off than the ELISAs.
Yeah, then I would definitely agree on that. Yes, I think
But
they
this
can
is
provide
like
you
more,
better.
I think, Dr. Sanjay, it is more closer to our heart, because probably, as
True.
you said, it was related to your PhD. My PhD was entirely on all the biomolecules assessed in follicular fluid and stuffs, and with ELISA. So very interesting, and it would be good to work on this, something related to this.
Yeah.
So any more questions, Nidhi?
So
I think there's one Dr. Manish Garg has
yeah,
asked.
Dr. Manish. Dr. Manish, you can directly ask the question
you can
if you're
ask.
there.
Sure. Just was wondering if nutraceuticals like resveratrol and melatonin have any health benefits through these extracellular vesicles. I was just doing a literature search and there was some preliminary data, but I thought I'll take views from the speakers.
Yeah, can I answer it? Like, or Dr. Ricardo, if you want to go ahead, like, please.
No. You are more entitled than me to answer.
Yeah, I think we're currently doing actually one mock study on this, but not melatonin or resveratrol, but for progesterone. Looking at how the EVs varied on the of different phases of the menstrual cycle. We're looking at the progesterone level at the same time, or the EV production level. So of our, like, it's still preliminary data that we have. So we see an influence of the progesterone, how it is improving the EV production itself, like in a higher range. But yeah, to say, I know some bovine studies were done on the melatonin, where it regulates actually a lot on the embryo implantation. So it's particularly when they are on the stress conditions. Look, melatonin is highly used. For in bovine studies, they use on the heat stress when they're on the heat in the summer and winter months, when they're on the summer months, they used melatonin as a treatment, and they also extracted EVs from the follicular fluid, and they saw that this melatonin supplementation as an antioxidant improved the EV production itself, and it has a positive effect actually. But I think on the human level, I don't know how the research is going on. I have no idea yet, but I still need to see that.
But progesterone makes more sense, not only for the female, but in terms of males also, because it serves as a chemoattractant for the sperm motility as well. So
yeah.
So,
Thank you, sir. So
Chaitanya,
you have-
can I check? You said positive effects of melatonin on what outcomes?
Melatonin in terms of reducing the stress, I think it was regulating some of the stress factors, particularly the heat shock proteins that was involved actually. I can send you this article if you can send me an email. I have this I read this article from Pete Hansen's group from University of Florida. He worked a lot on melatonin, also on the heat stress factors. There are two papers published from Colorado group and also from Pete Hansen's group. But this is again bovine studies that I'm talking on. But they did work specifically EV derived, and these EVs cargo content has an influence on the heat shock proteins.
Sure, I'll do reach out to you. Thanks a lot.
Yeah, sure.
I think it is HSP 60 and 90 probably.
Yeah, yeah.
Thank you so
So
much.
it's over to you,
As
Nidhi.
you have likely said, Sanjay sir, sir has been emphasizing all what you have said. We have to really study hard in the human field now. I think so. We have to grossly go into it. And again, thank you so much for the wonderful presentation. And I don't think so, sir. It was shortened at any level from Ricardo sir's presentation. It was equal enlightening and thought-provoking, I would say. And Sanjay sir, if you'd like to say something, and Ved sir, if you could conclude.
I'm speechless.
Yes, yeah, yes, sir. Ved sir, if you could conclude and the
So
webinar.
thank you, Nidhi, and it was wonderful session. Actually, I heard so many things. I couldn't join in between, but it was wonderful. And I heard Ricardo and Pawnee. I heard of Pawnee earlier also in our two conferences. So they both are doing very good work. And when Dr. Sanjay Shukla reached out me to why not to do webinar on extracellular vesicle. So in 2 days before, I got one article from Dr. Ricardo from that, that LinkedIn. So I told Dr. Sanjay, why not to call Dr. Ricardo also, because that will be better, the combination, and we will learn so many things from them. So it was an excellent experience, and I think we hope that in future, definitely we will able to use this kind of new invention in our day-to-day practice. And thank you so much, each and everyone. Thank you. Thank you. Dr. Bindu, thank you so much.
thank you so much. I
Dr.
would
Bindu.
just say that's such interesting topics that we come up with. If we can really do something, I mean, we are enlightening ourselves, of course, but then if we can really do something practically, constructively, we can collaborate and do something probably also.
Right.
Ma'am, it is like apple has fallen on our head from the tree.
Yes. At
So
least
the small,
we have
to now think about what to do about it. Thank you so much to both our wonderful speakers. Thank you to both our moderators, and thank you, sir, for organizing this webinar. And here we conclude the webinar. And
thank you so much to that our UNS team and medical team, and special thanks to Dr. our Nakul Patil, who always agreed to for this kind of activity, and thanks to Dr. Manish also. Thank you so much. And of course, I forget the name, that Bhakti. She was coordinating me.
Thanks a lot, sir. Thank you. So nice of you.
Thank you so much. Thank you.
Thank you, sir. Thank you so much.
Thank you.